we're going to go with, get started with introductions anyway.
Is this your water?
Yes.
You recall in the news last week that President Biden announced no more Mr. Nice Guy. Everybody's going to have to get vaccinated who is employed in a place that has 100 or more employees. Healthcare workers are going to be required to do so. And I think government employees and all sorts of things. So it's the type of thing that gets a lot of different reactions, and I've noted here in reading the Wall Street Journal that the South Carolina governor, whose name is Henry McMaster, tweeted this in response to the Biden plan. Rest assured, we will fight them to the gates of hell to protect the liberty and livelihood of every South Carolinian. And then the governor of Georgia said, I will pursue every legal option available to the state of Georgia to stop this unlawful overreach. So some of you might stand up and say bravo to those two governors. Others might boo.
No, that's okay.
The critical thing is, before we react, we keep gathering information and get to the truth. And as Dr. Oxford said today, we want wisdom, and we can listen to the cries in the streets as well as the cries of politicians. Here to make sense of it all, I think it's the third time, John.
It's the third time.
Is a COVID update from Dr. John Russell, who most notably is a colleague of Dr. Fauci. John is a scientist, educator, and a member of this congregation, has been wonderful in helping us to understand the issues of the pandemic. Thank you, John.
Thanks, Richard. How many people are tired of COVID? And I was really excited this spring as the vaccines rolled out that maybe we really were going to be over COVID. In fact, Carol and I went on a cruise, an all-vaccinated cruise to Iceland at the end of June, and it looked like things were really going well. And then all of a sudden in July, everything went down the tubes because of this new variant, Delta. So I thought I'd talk a little bit about some background and then why the challenges are so great still. So first we're going to do a little bit of basic cell biology, that you need to know the cell biology to understand the virus infection, the immune response, and vaccines. I'm going to try to make it as simple as I can, but don't hesitate to ask questions. And we're also going to talk about some of the scientific basis for some of the misinformation that's out there. There's a lot of misinformation that actually has a scientific basis, but it's just kind of half-truths. The one I'm not going to talk about is injecting chips. There's just no scientific basis for that. And we'll talk about how the different vaccines differ, and then what are the variants, or what are variants, and why are they creating these challenges. So the basic cell biology, I only want you to know two different pathways in the cell. One is what I'm going to call, for simplicity's sake, the garbage pathway, where the cell takes things up from its exterior, brings it inside, and then degrades it. And then I'm going to talk about the assembly pathway. And so the assembly pathway is how the cell makes proteins, which are the workhorse of what the cell does. They're enzymes that do chemical reactions, they're structural proteins, they're channels that allow ions to go back and forth. And here, it starts in the nucleus with the DNA. It has the code to make the protein. In the nucleus, it gets transferred into RNA, which then comes out into the cytoplasm, and then the protein gets made based on that RNA sequence. And what the gene really is the sequence of how you string together different amino acids to make an individual protein. So amino acids are things, they're the supplements. Methionine, arginine, leucine, you can get them in supplement stores. So there are 20 of them, and each protein has a unique way to string those 20 different amino acids together. So that's the assembly pathway. And I'll talk about a protease. I'll talk about proteases, which are just proteins that break down other proteins, that cleave and chop up proteins. So, are we straight so far? Assembly pathway, garbage pathway. That's all you have to know, assembly and garbage. So this is the structure, a stylized structure of our friend the coronavirus. And these purple representations are the famous spike protein. And that's the protein that is the target of all the vaccines. And it's the protein that allows the virus to stick to the cell and start to invade. So let's talk a little bit how that happens, how the virus gets in. And the first thing that happens is that spike protein binds to another protein on the surface of the cell. In this case, it's called the ACE2 receptor. And the virus binds to that protein on the surface of the cell, and then it enters the garbage pathway. So it goes in with one of these vesicles, and the problem that the virus has is it's surrounded by membrane, and the cell is surrounded by membrane. And so if the virus is going to work, it has to unload that RNA that's inside the virus and get that inside the cell. And so the garbage pathway allows that to happen because as the virus moves in, it's encapsulated by cellular membrane, and it undergoes an acidification process so that the inside of this, where the virus is, becomes more acidic, like very dilute vinegar type solution. And when that happens, it changes the structure of the protein, and the protein gets attacked by a protease that's inside this vesicle, and that allows the virus to fuse and dump the RNA inside the cell. And then once the RNA is in the cell, it goes into the assembly pathway and uses the machinery inside the cell to make viral proteins, to make new virus. And this acidification process is actually inhibited by chloroquine. This had been shown back in the 1970s that you could show in a dish these RNA viruses. You could prevent them from infecting a cell by putting them in high concentrations of chloroquine or hydroxychloroquine. Unfortunately, you can't get the concentration inside a person to mimic what happens in the dish to prevent the entry of the virus. Some of you may even take hydroxychloroquine and not realize it. The generic name for it is Plaquenil. It's also used for rheumatoid arthritis, and we'll talk about that in a minute. Now, one of the reasons that this virus, this SARS-CoV-2, is so different from the others is that it figured out a different way to get inside the cell so it doesn't go through the garbage pathway. It mutated the spike protein so that it has another site in that protein that's called the furin site. And there are many, which is creating another site for an enzyme to cleave it. And in this case, it's this beast here, but we won't worry about that. But what happens is that opens up the virus right directly on the surface of the cell so that it can dump that RNA without going through this pathway. And that's what makes this virus so special. And now I'm going to get a little geeky because I'm going to explain what that is and why it's caused controversy. So these are the furin sites. These are sequences of amino acids, and this is the sequence at the furin site of the COVID-2 virus. And it's these red R's separated by an A and then a red R again. That is where the protease binds and chops that protein open so that the virus can then fuse. And what's different about this virus, we've had two other coronavirus epidemics since 2000. I don't know if you remember 2003, it was called SARS, and it's now called the COVID-1 virus. And it didn't have a functional furin site, so it had to go through the garbage pathway. In 2012, there was the camel virus, the Middle Eastern respiratory syndrome virus. It also didn't have a functional furin site. So these two viruses, and we're going to talk about them more later and why they confused people at the beginning. But one of the things that's happened, one of the controversies about the source of the virus is where did the COVID that we have now, COVID-2, where did it get this furin site that makes it so infectious? And people looked at the original COVID virus that came out of China, and it was very different from this. So in this case, these amino acids are missing in the COVID-1 virus. And so people said, well, wait a minute, that's a pretty big change. Somebody must have engineered it. That couldn't have happened randomly. And that's where all of the controversy is with the origin of the current coronavirus, is how did it get so infectious? Was it a natural mutation, or did somebody make it? I think once you realize that there are other coronaviruses that are pretty close, that don't have to make the big jump from here to here, it's still possible that and I think most people think it's still likely that it came naturally from some animal host. But that is the source of the controversy. And the other thing that's happened, so this is the original COVID-2 virus. The Delta added another R amino acid that makes it an even better protease site, makes it even more infectious. So does that make sense why people were so worried about this furin site and how it contributed? Because that's really the source of the controversy. And, you know, it's perfectly possible that in Wuhan they were actually fooling around with this and they made this and it escaped. I think it's unlikely, but I don't think we can say absolutely it didn't happen.
John, can we find out ever?
It's really hard. I mean, what they really need to do, and the Chinese aren't helpful in this sense, is looking at what all the different viruses that they have in their repertoire, because they've been collecting these viruses for 10, 15 years, well, more than that, ever since COVID-1 went around. But that's the only way to know. But that's a source of a lot of controversy. So now we go to the immune system. And we go back to the assembly pathway and the garbage pathway. And we do that because the immune system is trained to look for differences in what's normal versus a virus coming in. And the T killer cell looks for changes that occur in the assembly pathway. So the assembly pathway, when it makes new proteins, it's kind of sloppy. And bits and pieces of those proteins get chopped up, and they end up on the surface of the cell. And the T killer cell can tell whether that's a normal protein or whether it's coming from a virus protein that it's never seen before, and it attacks it. The other T cell is the helper cell. It scans the garbage pathway for changes. So these two T cells are the sort of the engine to decide what's self and not self and what we should attack and what we shouldn't. And again, it's the assembly pathway and the garbage pathway that they're looking at. So if we look at the T killer cell, very near and dear to my heart, that's what I started my research on, it attacks, so this is a target cell. Here's the little killer T cell. Here it's bound the target cell. And within minutes, this cell, when we were doing these experiments, I was reading Shogun. And I said, you know, this is inviting seppuku. So what the killer cell does, it tells this cell to commit suicide. And it does. It also then stimulates inflammation in the region, secretes things. So that's the killer cell. And it's looking at changes in the assembly pathway. The B cell is the one that everybody looks for because it's the one that makes antibodies and secretes it. The B cell requires the helper cell to get going. But the B cell makes antibodies, this little Y-shaped protein here. And the goal for the B cell is to make an antibody to this spike protein so that the spike protein can't bind to the target protein on the surface of the cell. And these are called neutralizing antibodies. And they're relatively easy to measure because they're in the blood. It's really hard to measure what the T cells are doing because it's a much harder assay and much less precise. But T cells can see things that B cells can't. So the good thing is, even though you may not have neutralizing antibodies, you may still have killer T cells that can help with the response. So what does an immune response look like? So the immune response, this is divided into 3 phases. There's an early phase, right after infection or right after you give the vaccine. And there are proteins and cells that are kind of poised to just look for things that are different. Not very specific, but things that are different. And that's called the innate immune response. And that's good because it turns on within hours, but it has a very low capacity. So if you get a lot of virus in, there's a limited amount that this innate immune system can deal with. What you need to do is get the adaptive immune response going. That's the T cells and the B cells. And that takes a while because the number of T cells or B cells that are reacting to any new infection is low. 1 in 100,000 or 1 in a million. That's not enough. So what happens is at that initial infection, those cells start dividing and becoming activated. And that takes generally a week or 2 to reach a peak. And that's called the adaptive immune response. But once those cells get going, they have a much larger capacity. So they can handle a lot more virus or a lot more bacteria. So that's why it's really important in a vaccine to get these T and B cells going. And then once you start that primary response, it sort of peaks within a week, between 1 and 2 weeks, and then those cells sort of quiet down because the challenge is gone, there's no virus there, and they become memory cells. And what happens then is the next time, so when you get the booster shot, they are ready to go and they blast off and go and start dividing even more. But it's really critical that these cells have this little rest in between because these cells, while they're dividing and activating, they aren't very responsive. Another stimulus actually can kill them rather than stimulating them to grow. So this rest period is important. But it's really arbitrary. I think Pfizer, they had 3 weeks. As an immunologist, you wouldn't do 3 weeks. You would do them 4 to 6 weeks before you came back. But Pfizer wanted to get through those clinical trials as quickly as they could, so they cut it as much as they could. Moderna said, well, we're going to do 4 weeks. And some of the other ones said 4 weeks too. But this is really a little bit arbitrary. You just need to make sure that they have a little time to rest so that they can respond again. And actually, the Brits, they kind of did a clinical trial within the response. So they were worried. So there was a policy issue. When you had a limited amount of vaccine, what do you want to do? Do you want to cover as many people as quickly as you can, or do you want to make sure what? Oh, sorry. Question?
Yeah, I just wondered, when you were pointing to that third one, are you referring to what we're calling the booster, or are you referring to the second shot?
It's a second shot, but the booster will do the same thing. The third shot will do the same. The third shot's given over here, it does the same thing. So back in the spring, or last fall into the spring, there was a lot of discussion of what do we want to do? Do we want to cover as many people as quickly as we can, or do we want to get as high a quality response in fewer people, and take the chance that that they may not be optimally immunized, and therefore they may provide a source for variants, escape variants, or do we want to cover as many people as we can? And the Brits, within their vaccine program, some of the people they allowed to go 12 weeks instead of 4 weeks. And when they looked at those people, the antibody titers were 1,000-fold higher after they waited 12 weeks rather than 4 weeks. So that was a policy decision that they made. And so now, as you can see, after the second booster, it still decays, but now you come back with another booster, it's going to take off again within a couple of days. So that's what the discussion now is. Should we do that or not? Okay, so now let's talk about variants. So this is the structure, and the neutralizing antibodies. So this is sort of a description of the structure of the spike protein, and it's actually made of 3 identical subunits stuck together. And then this is the region of the protein that binds to the ACE2 receptor on the cell. And so the issue is that the concern has always been that what you're trying to do with the antibody is you're trying to get antibodies to this part of the protein so that it can't bind to the cell and cause disease. Well, of course, the virus is mutating as you're doing this, and the virus wants to become more and more infectious. The more infectious viruses, it's an evolutionary thing. The more infectious viruses win out over less infectious viruses. And so the worry has always been that the virus is going to mutate this binding site. And as it does that, you may lose the neutralizing capacity of the antibody. So far that has not been a problem. And we'll talk about that more in a minute. Okay. So now let's go to vaccines. It turns out that when COVID hit, vaccine people were they had developed whole new technologies beginning in the mid-90s right up until 2019 that were ready to go. And one of these technologies were these adenovirus vectors. So these are DNA viruses that cause common colds. And so AstraZeneca had been working on this, and one of the problems that we've had during the development of these adenovirus vectors is that they're a human cold virus, and so everybody's been infected with them. And so the problem was finding an adenovirus vector that wouldn't just be cleared and not be effective because you'd already been immunized with it, it wouldn't infect and provide immunization. So AstraZeneca had been doing this, and to get around it, they used a chimp virus. So the human had never seen this chimp virus, but the chimp virus would infect human cells. And they were getting ready to roll this out as an Ebola vaccine. So they had the vector ready to go, but they were going to, the clinical trial was going to be in Africa for Ebola. But what these adenoviruses do is they've stripped the insides out of the adenovirus. Instead of having the adenovirus DNA in there, they put the DNA for the spike protein in there. So that the adenovirus essentially becomes just a DNA vaccine, a way to get DNA into cells. And so J&J used a very rare human virus, adenovirus as their vector. And the Russians, they were so worried about this business of the vector creating an immune response and making second injections useless, they used 2 different viruses for the first and second dose. But they're all good, and all were involved in just getting as we'll talk about, getting the DNA for the spike protein into cells so that you can make a response against it. Oh, and one of the problems that still happens is that these adenovirus vectors are grown in human cells. I mean, they have to bind human cells. That's how they replicate. You can't use monkey cells or something else because you want to get something that's going to recognize human cells. Well, some of the cell lines that were used to grow the adenovirus vectors came from aborted tissue. And so there was the ethical concern about using these adenovirus vectors because of the aborted tissue. The other technology that had begun in the 90s was to make an RNA vaccine, get away from the DNA, and get away from virus vectors to get the stuff in. Just make an RNA to the spike protein and somehow get that into cells. And that started in the 90s. And it went through a number of iterations, and there were problems that were not solved until about 2015 because the RNA was causing inflammation. But by 2019, those problems had been solved, and what happened was warp speed. The great thing about warp speed is all this technology was there, just waiting for somebody to give it some money to explode and see whether it would really work or not. And that's what happened. So the good thing about warp speed was dump money, particularly to Moderna, which was a kind of a startup company at the time, and they got a lot. Interestingly, Pfizer didn't accept any warp speed money because they didn't want to lose the intellectual property. So they developed it on their own. And that's been an issue now going forward as you try to ramp up more to get more global supplies. So Pfizer didn't want to give up their intellectual property. The other vaccines basically use tried and true methods. So Novavax actually makes the spike protein in insect cells, purifies the spike protein, puts some adjuvant in it to stimulate inflammation, and then injects that, and you make antibodies directly to the spike protein. So the spike protein's going into the garbage pathway, so you're not getting any T killer cells out of that. Okay, so here's the way the viruses work. The adenoviruses, they infect the cell, they are engineered to put large numbers of copies of the spike protein into the nucleus of the infected cell. Then just using the assembly pathway that the cell already has, it makes RNA from that DNA, pumps the RNA out to the spike protein RNA out to the cytoplasm, and it gets made into protein. In contrast, the Pfizer and the Moderna, they just inject the spike protein RNA directly into the cell, and it makes the spike protein, and that's where the immunization comes from. And there's a lot of talk about, you're messing with the DNA of the nucleus and all. The RNA never goes to the nucleus. It goes directly into the cytoplasm, into the assembly pathway, and makes the spike protein. So the comparisons of the different vaccines. So Pfizer and Moderna, they are the RNA vaccines. Their biggest disadvantage is storing them to make them widely available. When Pfizer first came out, it had to be at minus 80, stored at minus 80. And that's because if you're in the lab, that's the way you store viruses. You just do it that way. And rather than fool around and figure out whether it really needed to be at minus 80, they just said, we're going to do that. Moderna fooled around and got some biomaterial people involved, and they found a way to stabilize the RNA so it didn't have to be stored. It could be stored at or freezer temperature, normal freezer temperature. The J&J vaccine, it's been star-crossed. I mean, it's a good vaccine, but there were so many issues in when it came out. It was a single dose, easy storage. It made T killer cells and antibody, but there were just so many problems. First, it was the blood clots, and then the facility in Baltimore that was making it got contaminated, and they had to throw everything away. If they ever solve the problem and people can forget about the problems that they had bringing it out, it's still a great vaccine. AstraZeneca is another adenovirus vaccine, great vaccine. Again, it's still an issue to see what happens about whether you make antibodies to limit the efficacy of continued immunization with these adenovirus vaccines. Novavax, we talked about, is just making the purified protein and injecting that. The Chinese virus, the Sinovac, that's old technology. They just grew up a bunch of COVID virus, killed it, and inject that. And as a result, it's not all that effective. It's like a flu virus, or a flu vaccine. Okay, so we talked about the recent outbreaks. So there were two previous coronavirus outbreaks. As I said, the SARS-CoV-1 in China, 2003, and then the MERS-CoV in 2012. And what happened with both of those outbreaks is they just disappeared. The viruses were not that infective, and they just disappeared. So CoV-1, fewer than 10,000 cases worldwide, but they had high mortality in the cases that got infected, but the virus was not very effective, and basically the Chinese got rid of it by masking and social distancing, because there were attempts to make vaccines against CoV-1, and they all failed. The vaccines that they made actually made the virus more infectious. And so that put everybody way behind when COVID-2 came out, because they were worried, well, how are we going to make a vaccine? And then also, the gain of function. How many have heard gain of function? No? Okay. Gain of function was when we were talking about taking the SARS-CoV-1 and putting the furin site in it. That's a gain of function. You gave the virus some function that it didn't have before to make it more infectious. Well, Chinese and the Americans and the Swedes and the Swiss and everybody else, one of the ways that they figured out that the SARS came from a bat was that they reverse engineered the bat virus so that it bound to the human receptor. And so that was what convinced people that, that's probably where it came from, because the bat virus would infect human cells if you just changed that piece that recognized the receptor. And again, the MERS-CoV in 2012, there were fewer than 3,000 cases worldwide. It just died out. And I know that's what Trump was hearing. People were telling him that's what happened to these earlier things. And, he said, well, we got 15 cases today and they're going to be 0 tomorrow. That's what everybody was relying on. But the epidemiologists knew that wasn't happening. I mean, when the Chinese canceled Chinese New Year in February of 2020, it was clear they knew, and then everybody else knew, this is not grandma's COVID virus. This is something totally different. And then the Americans, for reasons that I still don't understand, CDC really let us down. We didn't test enough, or we didn't test at all, because the CDC insisted that we use their test. And for the critical 3 weeks in January, February, the tests that they were sending out to use were no good. They were contaminated. So we were flying blind until March. And once we got behind with this virus that was so infectious, we're still trying to catch up. And now with the Delta, we're trying to catch up again. And that's what happened this summer. As I said at the beginning, I was really optimistic, May and June, that things were really going to turn around, that we were going to be able to go to Greece in October. Not happening. And the critical event for me, and I think for a lot of people, how many have heard about the outbreak at Provincetown over 4th of July? No? So, 4th of July, Provincetown, Massachusetts, 75% of the people are vaccinated. And suddenly, they find asymptomatic vaccinated people transmitting the virus. That was a game changer. And the reason that it was happening is when they did the nose swabs, they found that vaccinated people had the same amount of virus in their nose as unvaccinated people who were sick. So that's why we can't get rid of these. And so the Delta really changed the game and put us back. Hopefully we can come out of it, but it's got to be vaccination. That's the vaccination and social distancing and masking are the only arms that we have in the armory for these viruses. So if we look, this is again a picture of the virus, and this is the furin site down here, the protease site, and up here is the receptor binding domain. And the mutations in Delta that are really important appear to be this arginine that got put into the furin site that made it better. But it looks like this mutation up here in the receptor binding region is more important. And that's based on cell infection assays. The Delta is a better substrate for the furin. But this mutation right here, where it's mutating a leucine to an arginine, that stabilizes the binding to the cell and makes it much more infectious. That's why it's 1,000-fold more viral load with COVID Delta than with COVID Alpha. Okay, so where are we and what are the challenges and concerns? To me, the Delta is the viral loads, it's 1,000 times more than the original COVID-2 Alpha. Now there's Mu and Lambda and everything else out there. And the longer Delta stays around, it'll make more mutations that could be even worse. But the last I saw, the Lambda and Mu, they're not nearly as infectious as Delta. And so just from a viral evolution, they don't look like they're going to be able to take over. Maybe they will, but Delta is the one. And so the other thing that people are really worried about now is we're headed into fall. This is respiratory virus season. And the problem that they're seeing with kids is that they're getting doubly infected with COVID-19 and respiratory syncytial virus. And these kids are really sick. And now, with flu coming, I got my flu shot on Wednesday, I think it was. The rest of us, even though we're protected pretty well if you've been vaccinated, you could be asymptomatic for COVID-19 and get the flu and make everything a lot worse. And I'm on a board, and they said, well, when can we go back? And I said, to me, in-person meetings, I said, not until my grandkids get vaccinated. That is the real problem right now, is as the kids go back to school, they aren't vaccinated, they can get the virus, they can transmit it. It's really a scary time until more people until everybody can be vaccinated. And then better drugs. Hopefully there's always ivermectin out there. I was going to talk about ivermectin. Ivermectin is a great drug. It's a great drug for parasitic worm infections. It binds to, it basically paralyzes worm muscles. And we don't have the protein that it interacts with in our muscles. We have it in our brain. That's the problem. And normally, ivermectin is kept out of the brain. But if it gets in the brain, that's a problem. And so the basis of using ivermectin comes from, again, some studies in tissue culture in Australia in 2020. They showed that ivermectin could block COVID infection of cells in a dish. Unfortunately, it was 100-fold higher concentration of ivermectin than is used clinically to treat worm infections. And at that level, it's going to get into the brain, it's going to cause, that's what happens, the toxicity of ivermectin are CNS toxicities primarily. And then if you're using horse ivermectin, it's just not formulated the same way. You don't know what else is in there besides the ivermectin. So, but the NIH actually started clinical trials in June on ivermectin. There are studies out there that say it's effective. Most of those studies haven't been confirmed, or there's problems with the data. And Merck, the other thing about ivermectin is it is such a good drug. It has made such a difference in the third world with parasitic diseases. The 3 Merck scientists that found it and developed it got the Nobel Prize in 2015. So it's a great drug. It's just not a drug for COVID, at least not now. Maybe something else will come up. I know Pfizer and some of the other companies are working on protease inhibitors that block viral, basically based on the same kind of thing that they do with HIV. Whether they'll work or not, I don't know. But vaccination is clearly the way that we get out of this, at least in my estimation, in the short and long term. Richard?
John, our grandkids, are some of them going through clinical trials right now?
Somebody's are, not mine, but no, I think.
Actually that far along?
Yeah, Pfizer, I think, thinks that they'll be ready to apply for emergency use for, I think, 2 to 12 in a few weeks. They actually thought they were going to get there earlier, but the FDA asked them in June to increase the number of trial subjects. The FDA is really overly cautious to make sure that there are no safety issues. Not that many kids get that sick, so they just felt that there weren't enough kids in the trial, and they asked them to add more. But I think Pfizer just announced that they're ready in a couple of weeks, I hope, to ask for emergency use authorization. And there are people who are saying that, come on, FDA, give us a break. But they don't want to be accused of being politically, they want to make sure that the science is right, and so they're going to make them wait. But it'll probably be Christmas or so before that kids can get vaccinated.
Even with the emergency approval?
Yeah.
This last week in the science.
Huh?
This last week in the science section of the New York Times, it said that at the end of September for Pfizer and 2 to 3 weeks later for Moderna.
For Moderna, yeah. For the emergency use authorization, right. And then it'll take time to roll it out, and then, there'll be people that say, no, you're not vaccinating my kid, but until we get more people vaccinated, we're going to be in this mess for a while. Yeah, Skip.
I've heard some talk about combining the vaccination with the flu shot this fall. Is that good or bad?
I think it's good news. Some people are scared of it, but I think it's good. I know Novavax, the one that makes the protein, they're already getting ready to do it. And Moderna has announced that they're going to have an RNA vaccine that's going to do both flu and COVID. Yeah.
I got my Moderna last shot in February, because I was in the early group. And what is your opinion of the booster and when you should get it? You know, they say 8 months.
I would say get it as soon as you can. It's really arbitrary. You know, when they had this discussion between the first and second, Tony kept saying, you know, this is the protocol that we've done. We're not going to change the protocol. The Brits said, we're going to, the heck with the protocol. We think it'll work because we're trying to cover more people. And it turned out that they did. And the other thing that comes up is, does it matter what you get the booster with? No. 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 Get a booster with whatever you can. The immune cells don't know whether that RNA or that protein got out there by way of an RNA virus or an RNA vaccine versus an adenovirus vaccine. If you can get a booster, get it. And I was asking Pam earlier, I don't know why Pfizer isn't doing, coming out, instead of getting a regular booster, get a Delta booster. The advantage of the RNA vaccines is they're pretty nimble. You can change from one to the other very quickly, whereas the adenovirus or the protein vaccines, it's a much bigger manufacturing process. But with the RNA vaccines, they should be pretty nimble. They could go from one sequence to the next pretty quickly. But Pam said she thinks that the government was going to make them go through full phase 3 testing again, just changing a couple of residues in the RNA. Yeah.
Do you see the RNA vaccines being the vaccines of the future?
Absolutely. Absolutely. And again, because they are so adaptable. The problem with the flu is you're always fighting last year's war. And these things, you can wait till you really know what's going on and then make a much more specific vaccine.
Do you have to go through another trial or anything like that when you make the tweak to the RNA?
That's what Pam thought that they were going to make them do. And, maybe because it's new that they would do that. It doesn't make any sense to me, but it could be. I mean, they don't make people go other than a few safety trials when they change the flu vaccine. They don't make them go through a complete clinical trial. But yeah, I think the RNA is clearly a...
Professionally, what is your role in all this? Individually, John Russell, are you involved in?
No. Those pictures of the killer cell killing the target, that was in one of the first papers that I published back in 1982.
Oh, you mentioned that.
Yeah.
That was way back when.
Yeah, 1982, yeah. But no, I don't have a lab anymore. I've just been interested in this and the whole subject and trying to make it up.
Giving this presentation tomorrow at work?
Well, it's for it's our Society of Professors Emeriti.
Oh, okay. Very good.
Bill got me roped into that.
Hey, John, is there another speaker series presentation on how you guys know this stuff? I mean, when you say, well, we see these cells and we can tell the assembly pathway and the garbage pathway, how the hell do you know all that stuff?
Oh, that's...
Pretty elementary?
No, that's, really... Some of that, Mechnikov, goes back to Mechnikov in the 1890s. I mean, this idea of complementary proteins interacting with each other, that's 1890 or something like that.
No.
But I mean, the cell biology, most of the cell biology is within the last 50, 60 years.
You look at it through a microscope.
No, I mean, most of the stuff is done in tissue culture and with animals, and then you go to the human. And...
I want to know. This is great. Anything else? Thank you, John. And here for all of your good work. is a unique cup that I found that says,
Dr. Fauci, we trust, yes.
"In Fauci we trust."
Thank
you, John.
I should have worn my Fauci t-shirt.
Thank you, John.
Yeah.
What does it say?
In Fauci we trust.
I say we need one Fauci for president.
Yeah. I've got one. I've got a hat.
There was one that had Fauci on one side and Trump on the other, truth and fiction. And I won't say which one was which.
Jim, thank you. That was very good.
By the way, we did have up to 5 people following us on Facebook, so they figured it out.
You still on?
You done?
Yep.